Certain microorganisms are identified in the lab via molecular techniques only. For example, gonorrhea and chlamydia infections utilize nucleic acid amplification tests, while salmonella can be detected using a DNA probe. In this lecture, we're going to investigate what is meant by a DNA probe and PCR. Restriction enzymes. So restriction enzymes, also known as restriction endonucleases, are enzymes that can recognize a particular sequence of DNA and cut every time that it sees that sequence. So for example, if this restriction enzyme recognizes ATTC, and you have a strand of DNA that says something like GATTTCACCATTC. The restriction enzyme is going to scan the DNA and read ATTC and cut the DNA. Then it's going to keep scanning, and then it's going to read ATTC and it's going to cut the DNA. And since DNA is double-stranded, it's also going to read in the opposite direction and cut any other ATTC sequences that it sees in a row. So anytime it sees that particular sequence, it's going to cut the DNA. Restriction enzymes originally came from bacterial cells. The reason why bacterial cells produce these is because of those bacteriophages that I mentioned previously. So a bacteriophage is a virus that can infect a bacterial cell. And when a bacteriophage tries to make a bacterial cell sick, what it does is it injects in viral DNA into the bacterial cell. Well, this incoming viral DNA can kill the bacteria. So what the bacteria does is it produces these restriction enzymes to kill off the viral DNA. Well, we figured this out, and we thought these were pretty cool because they only cut at particular sequences. So we can cut, mix, and match DNA now because of these restriction enzymes. So once we cut the DNA, then we usually run it out on a gel. So gel electrophoresis. Gel electrophoresis is a mechanism used to separate strands of DNA by charge and size. So there's an electric gradient, and going back to the picture of this gel, there is a negative charge that's imposed on this end and a positive charge that's imposed on this end. DNA has an overall negative charge. So in these wells up here, and here you can see a diagram of the wells, and this is an actual picture of wells. So in these wells, you load the DNA. And this is all in a buffer solution system. And then you turn this machine on, and a negative charge will start to be generated on this end, and a positive charge will start to be generated on this end. Well, because the negatively charged DNA is on this end, it's actually going to be repelled away from this negatively charged area. So DNA is going to try to get away from the negative charge, and it's going to be attracted to the positive charge. So it's going to migrate toward the positive charge. One of the easy ways to remember this is we always say run to red. Red is usually the positively charged node there, so run to red, the DNA will go through the gel toward the positively charged electrode. Now with DNA, the smaller the size band, the faster it can move through this gel material. So larger bands are going to be higher up in the gel, and smaller bands make their way through faster. So DNA passes through the agarose gel, small fragments move the fastest, and then it leaves a readable pattern of DNA fragments. We can use this for a lot of different techniques, and in molecular diagnostics you talk about a lot of those techniques. But I'll just give you a quick example of a paternity test. So how do we know who the father is? Well, we usually load moms, babies, and then any potential fathers. So mom, baby, dad 1, dad 2, dad 3. And you can do this for 100 dads. Hopefully there's not that many possibilities, but you never know. So when you load moms, babies, and dad's DNA, the bands are going to start to migrate through the gel. So moms may have bands here, here, and here. Babies may have bands here, 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 and here. And dad 1 may have a band here, here, here. Dad 2 may have a band here, here, here. And dad 3 may have a band here, here, and here. So now we have to analyze this to figure out who the father is. Well, the baby gets 1 chromosome from mom and 1 chromosome from dad. So what the baby ends up with is banding patterns that partially come from the mom and partially come from the dad. So first, since we know who the mom is usually because the baby is coming out of the mom, we can cross out any bands associated with the mom. So this band came from the mom, and this bottom band came from the mom. So that means that these remaining 2 baby bands had to have come from the dad. So let's look at dad 1. If we look at dad 1 did not contribute a band here, but it did contribute this band. But since there's 2 bands that have to be accounted for by the dad 1 could not be the father. Now let's look at dad 2. So dad 2 has a band here, so that's a good sign. And the other band is also accounted for by dad 2. So dad 2 is our likely father. Now if we look at dad 3 is missing this band, which had to have come from dad. So dad 3 can also be ruled out as the father. So our 99.9% certainty is going to be with dad 2. DNA sequencing. So all you need to know about DNA sequencing is it's getting the order of bases in a strand of DNA. Now, knowing the sequence of a strand of DNA can be helpful because we can identify particular genes that may be associated with drug resistance. We can detect particular mutations that may be associated with disease progression. So sequencing DNA and getting the exact order of bases allows us to look for those types of mutations or those types of genes that might be present. There's a lot of different DNA sequencing methods, and the new diagnostic methods are going toward whole genome sequencing, so that not only are patients getting the species of microorganism that's causing an infection, but now they're trying to do whole genome sequencing of the microorganism to know, well, what genes are present in that microorganism and how could those genes be affecting the overall health of the patient. The other thing that they're starting to do is do whole genome sequencing of a person. So the Human Genome Project was a huge, multiple-year project that finally got the entire genetic code, so the entire sequence of the entire human genome. And that Human Genome Project took many years, and now they're able to do whole genome sequencing of a person within a couple days. So the technology is speeding up here. But this is just an example of one type of DNA sequencing method. So it'll sequence and then incorporate these dideoxynucleic acids or dideoxynucleotides, which stops replication at that point. And then when you run them out on a gel and use particular detectors for the different amino acids, different nucleic acid bases, then the shortest band will have the first base in a sequence. So then you read from the bottom to the top. So T, G, C, G, T, this is the actual sequence of the DNA. Now, polymerase chain reaction. Polymerase chain reaction is widely used. It can make many copies of a particular region of DNA. So if we have DNA here, we design a primer that will copy DNA in this direction, and we design another primer that will stick to the other strand of DNA and copy DNA in this region. Now these primers are going to move toward each other and fall off, and through multiple cycles of this, you will make hundreds and hundreds and hundreds of copies of this particular region of DNA that exists between the two primers. And there's a video that I've posted on Blackboard that talks about how this is amplified. Now this region can then be identified on gel electrophoresis. So some applications of this. Let's say we have the MEK-A gene for methicillin resistance for Staphylococcus aureus. If we get the patient's bacterial DNA isolated, we put these primers on here that will stick to and recognize flanking regions of this MEK-A gene. It'll make tons and tons of copies only if it's present. So if you run this out on a gel and there's nothing there, then the patient does not have MRSA. But if you run it out on a gel and you do see a band in the particular size range, then the patient is positive for MRSA. So this is one application of PCR. Another application of PCR is being able to amplify small fragments of DNA that may be left at a crime scene. So even if there's a tiny, tiny amount, you can make tons and tons of copies through this amplification mechanism, and you will be able to identify who the criminal was. So the three basic steps in the PCR cycle are denaturation, priming, and extension. So denaturation, the double-stranded DNA separates. Priming, also known as annealing, is when the primers stick to the strands of DNA. So you'll have a primer stick here and you'll have a primer stick here. And then extension, the primers will elongate. And then you keep repeating this over and over and over again until you make all of your copies of that particular region. Now, Taq polymerase is a special type of polymerase that can withstand really high temperatures, because in order to separate the strands of DNA artificially, we need to increase the temperature to 94 degrees Celsius. That's really hot, and most enzymes would denature or fall apart at that temperature. But Taq polymerase is a special kind of enzyme that can function at that high temperature. DNA probes. So DNA probes are small segments of DNA that have the ability to recognize and bind to particular sequences of DNA. So let's say we have our DNA, and let's say the sequence ACGA is only found in Salmonella. Now this sequence would have to be much larger, at least 20 bases long, so it would have to be much larger than what is here, but I'll just use this as an example. So now let's say we have a probe, and there's different probes that exist. There's a probe that has a fluorescent molecule, and it would have a complementary strand. Now this probe would be washed along all DNA, and because the sequence is complementary to this sequence, it would bind, and then you wash away any unbound probe, and then you expose this fluorescent molecule to some type of fluorescent detector, and then you'll be able to see fluorescence. A lot of times this is linked to a quencher, so possibly on the other side of this is a quencher molecule that dampens the fluorescent signal, and it would actually take a break in this probe in order to be able to visualize the fluorescence. DNA probes is something that is discussed in a little bit more detail in molecular diagnostics. And there's these molecular beacons that exist as well, that exist in like this hairpin structure. And as there's a quencher here and a fluorescence here. And as this thing binds, it gets far enough away from each other so that the quencher doesn't absorb all of the fluorescent signal. Either way, a detector will pick up the fluorescence if the sequence is present. So if we design a probe that will specifically bind to a sequence that is only found in, for example, Salmonella, and our sample has fluorescence, then we know that the probe has bound to our sample because our sample is Salmonella. Just to clarify a little bit about this quencher and fluorescence, because this might be confusing right now. So a quencher is going to absorb a fluorescent signal. So if this tries to fluoresce, the quencher is going to absorb it all. And when you're looking at this, you won't see anything glowing. But if this quencher gets broken away from the fluorescence, the quencher won't be able to absorb all the fluorescence. So now the fluorescence is able to be visually seen. It'll emit off and you'll be able to see it. Again, this is something that is discussed in a little bit more detail in molecular diagnostics. So probes exist for Salmonella, Campylobacter, Shigella, Chlamydia, and there's many other probes that exist. So some of the recombinant DNA strategies. One of the major strategies here is to obtain a target gene, insert it into a vector, introduce it into a host, express the gene product in the host, and this is the basis for transformation in research. This is how we make insulin and human growth hormone. So to go through these steps in a little bit more detail, we have human DNA. And in human DNA, we have the gene for insulin. Now, at the beginning of this lecture, I talked about restriction enzymes and how restriction enzymes can cut DNA. So we use restriction enzymes, and I'll abbreviate those RE, to cut the DNA. So we'll cut the DNA here and cut the DNA here. So now we have purified gene for insulin. Now we're going to take a plasmid vector and we're going to use those same restriction enzymes to cut open the plasmid. So when the plasmid gets cut open, it's an open ring structure. So we can glue the gene for insulin into our plasmid. So when we say we obtain the target gene, so we've isolated the gene for insulin from human DNA. We insert into a vector. Well, now we've shoved it inside of a plasmid vector. Now we have to introduce it into a host. And there's different ways to get a complete plasmid into a bacterial host cell. One of them is simply just by increasing the temperature through a process called heat shocking. And we're going to go through that in detail in lab, but basically you're going to put the organism on ice, and ice constricts, so the bacteria are going to shrivel down, and then you're going to very quickly put them in a really hot temperature, so 42 degrees Celsius, and at that hot temperature the bacterial cells will expand abruptly, and when they expand, the plasmid DNA can sneak in through the membrane. Then you put them back on ice, and the membrane constricts again, trapping the plasmid on the inside of the cell. Now that the plasmid is inside of the bacterial cell, now when this plasmid is copied, transcribed, and translated, this bacterial cell is going to be pumping out human insulin because it has the gene for insulin here. And we isolate that insulin, and that's what is sold to people that have insulin-dependent diabetes. So some of the key players, restriction enzymes, again, those are going to cut the DNA. The insert is the gene to be inserted into the new organism. The vector is the plasmid, so that's what we're going to use in order to get that whole gene inside of the new organism. And then competent cells. Competent just means that the cells are capable of taking up genetic material. Not every cell can do this. So a plasmid, again, is a small circular piece of DNA that contains non-essential genes. So they're just extra genes. They can help the microorganism survive a little bit better, but they are not essential for the life of the microorganism. And there's three major parts that have to be present on the plasmid. First of all, the origin of replication. That's just a place where you can initiate replication. If you can't make copies of the plasmid, then it's useless. The second is an antibiotic resistance gene. And the third is restriction sites, so places that you can cut the plasmid open. So looking at this plasmid, this is PUC19. It's a commonly used vector for transformation and research. All of these different restriction enzymes can cut in this gene. There's an origin of replication where we can begin replicating this thing. And then there's a gene for ampicillin resistance. So let's say we have bacterial cells in a tube. And then we have the tiny little plasmid DNA that have been made, and they're floating around in the tube next to the bacteria. Through the process of heat shocking, some of these will go into the bacterial cells while the rest of them stay out. Now we want to weigh, so at the end we have some bacterial cells that don't have the plasmid and some bacterial cells that do. In the end, we only want to grow the ones that actually got the plasmid. So how do we kill off the ones without the plasmid? Well, if this is in a tube, if we put ampicillin, the antibiotic, into the tube, ampicillin is an antibiotic. It will kill bacteria. The only bacteria it won't kill is anyone that has an ampicillin resistance gene. So the ones that got the plasmid have the ampicillin resistance gene, and those are the ones that are going to survive. So as soon as you add ampicillin to the media, anybody that didn't get the plasmid is going to be killed, and the one that did get the plasmid is going to be alive. So the purpose of this antibiotic resistance gene is to select for the microorganisms that actually got the plasmid.